Aloha again. Today we have a presentation by Anna Flores. Anna is a botany PhD student at UH presenting drought response across development in Hawaii's endemic prickly poppy, Puakala. Her research is conducted through a permit with the HETF. Please note the permits are required when entering these zones for research purposes. Anna, begin whenever you're ready.
Thanks so much, Serena. I'm really happy to be presenting this. I'm always excited to talk about Puakala in this project in particular. So thank you to everyone who came to hear me talk about this project. Like Serena said, this is a project that largely addresses environmental stress response across development in Puakala, and in particular, I'll talk about drought today. And so thank you for that land acknowledgement at the top. I'd just like to add that I in particular live and work in Hupua of Waikiki and the Moku of Kona and the Mokopuni of Oahu. And I'm always honored and privileged to be here in what is effectively unceded Kanaka Maoli land. So I'm really grateful to share this space with Hawaiians and share in their knowledge systems. Which brings me to this really wonderful species you could see here with these bright flowers and this big landscape. And this work on this particular endemic is something that I'm going to just break up into pieces, and we'll talk a little bit about conceptually functional traits and how we use that to study how plants vary across development, ontogeny, in particular, the developmental stages and how we measure variation across that. We'll talk about the research objectives. So what the questions I'm trying to answer with this research. And we'll talk about the experimental approach that I use to try and answer those questions and some key findings. At the end, I'll talk about some future directions for this particular research. So first, I should say this project is part of my dissertation research at UHM. I'm a botany PhD student here, like Serena said, and my main interests in plant ecology generally relate to how plants respond to environmental stress. I worked with invasives and salinity stress as an undergrad, and I came to UH to work with Casey Barton, who turned me on to exploring these questions about stress, but across development. And so the way to do that is by looking at function and how we use functional traits to measure this variation, how this response is characterized. And in functional ecology, the question that we ask is how do things work? You know, how is this mechanism working? What is this organism responding to the environment? And functional traits are the tools that we use to do that. They're physiological, morphological characteristics of an organism that influence the performance, growth, and survival. And they vary at different scales. They could be at the biological scale, often organismal, so the individual scale, across space and across time as well. Together, these can help us understand the mechanisms behind a lot of ecological processes. In my research, I focus on the individual, on how functional traits vary within an individual. And at this level, this can be attributed to a couple of things. Phenotypic plasticity, which is the ability of an organism to change its phenotype in response to its environment or some sort of external stimuli. And then ontogeny, which is the process of development in which plants go from seedlings to adults. An example of some of these variations with some Hawaiian plants is Ohia lehua, which has some pubescent or hairy leaves, and others the leaves are smooth. And this is believed to happen, the hairy variations believed to occur at higher elevations where there's more exposure to sunlight and it forms a protective layer on the surface of the leaves as opposed to the smooth ones. An example of how ontogeny varies in some Hawaiian plants is Acacia koa. We're all familiar with these two leaf types in koa. Juvenile leaflets look like this here at the top, and the mature phyllodes look like this here at the bottom. And this is important because these this is a very conspicuous way to mark the transition between maturity and juveniles. And sometimes in koa, you can even see the intermediate as it's transitioning. Which brings me to an important aspect of my research, which is what ontogeny is and how traits vary across these developmental stages. So when I refer to ontogeny, I refer to the genetically regulated phase change that marks how plants go from seedlings to juvenile, to vegetative mature, to reproductive mature. And these are often confused with age and size, but they're very different. Age is a measure of time in years, and size varies particularly to how plants physically change, and that has to do with, anatomically, meristematic tissue, how it develops. Ontogeny is something that is regulated by genes and their particular discrete stages. And so throughout a plant's age, it may transition through ontogenetic at different rates depending on the type of plant. And then across ontogeny, functional traits vary and responsible to some predictable changes as it goes through these stages. It changes in resource needs and acquisitions capabilities. And so we can find how traits vary in these transitional phases, when it transitions between phases. One of the ways that we measure that is by looking at the whole pattern, right? A trajectory. So this pattern of expression. And we do that by measuring traits at particular stages and then plotting it to look at what it looks like, to what the whole pattern looks like. And here we can see how throughout the plant's life, this particular trait varies. Studies on ontogeny are really neat because they give a really extended snapshot of how plants change, how plants change, how they use resources throughout their life. This graph here on the left shows secondary chemistry in aspen trees and how that changes across ontogeny. These dotted lines are particular stages. And you can see how in late ontogeny, there's less variation in how much this particular chemical is produced. On the right, we have koa again that shows how at these different stages, the phyllodes, which are the mature stage, and the leaves, how they respond differently to lower water availability. And that's important because this is something that we can use to predict how well koa, for example, might fare under an intense drought. In a forest that has predominantly mature trees, they have a better chance of doing well under an intense drought versus the juveniles in that particular forest. So this is sort of the bigger context of this research. And it brings me to the questions I sought to ask with this research. So first I wanted to know whether or not these big patterns, these whole plant life patterns, trajectories across ontogeny, whether or not they're plastic. So do they shift when they're exposed to particular stress or environmental factors? And I also wanted to know if they are consistent with the prediction that when plants are in early ontogeny to late ontogeny, they tend to shift toward more conservative growth strategies. So when plants are in early stages, they might grow fast and try to build more tissue to become established. And as they go to later stages, plants might be more conservative with their use of energy and shift to other priorities like reproduction. These are the two main questions. And the way that I am characterizing that is with a plot like this one on the right, where I have ontogeny on the bottom, on the x-axis, and the trait values on the y-axis. And each environment or each stressor, the trajectory, the trait response is put in these trajectories and these lines that show us the whole pattern. And that brings me to puakala and why puakala is such a great study subject for this. In studies of ontogeny, it's not all plants have distinct stages that you can identify morphologically, but puakala does. Puakala has very specific stages where you can see where it goes from juvenility to maturity. And more importantly, it reaches maturity in about 15 weeks. So it's totally manageable in a time frame for an experiment like this. It also has really conspicuous defense traits. It's prickled pretty much everywhere. Here on the bottom left is a flower bud that has prickles. It also oozes latex, so it's a very well-defended plant. It has waxy leaves, and this might be from being exposed to so much open light. And more importantly, there's already been a lot of work on this ontogenetic shifts in traits on Puakala done by Casey Barton and other lab members throughout the time. So we can use this information to plan this project. Puakala, interestingly, grows in so many different areas, and the general trend is it's just very versatile. So shrublands, low elevation, high elevation. It's very common in disturbed areas. On the Kona side of the Big Island, they can be found just on the roadside. If you're on Hawaii Belt Road, if you're driving slow enough, you can see the bright white flowers right there on the road. So it's very common in these highly disturbed areas. It flowers late summer to early fall, but that can be extended. And seed production varies, though each of these pods usually has thousands of seeds. In some cases, the pods have hundreds of seeds, and it's still plenty. So this brings me to my hypothesis and predictions for how Puakala might answer these questions. And the first one relates to how plants, how resource use has been studied in plants. And so far we know that, so far studies have shown that from early ontogeny to late ontogeny, plants are more conservative, shift to more conservative growth strategies. And that's what my prediction is for Puakala in this case, that it will be more acquisitive, so very fast growing, fast development of leaves early on in ontogeny, and then shift to slower, more conservative resource strategies where maybe growth is a little slower, and it might shift to other priorities like building defense tissue, building defense structures. I also believe that plasticity will decrease. So as the plant, as Puakala reaches later ontogeny, it'll be less plastic, so less responsive to environmental factors. And then with defenses, we know we have a lot of evidence of how defense traits vary across ontogeny. Meta-analysis in 2010 found that there was this very clear pattern of an increase in the levels of chemical defenses from the seedling stage all the way through to mature stage in woody plants and in herbs. And that's what I'm also predicting for Puakala, that prickles and latex will increase in production later in ontogeny. And I also think plasticity will decrease. The way I tested that is with the greenhouse experiment, which I love to do. In particular, I conducted 2 greenhouse experiments, and in the first, we exposed Puakala to 2 factors that can shift across ontogeny, across the entire, across a plant's life. It can shift multiple times, the availability of light and water. And the way the design worked is I exposed Puakala to either low or high light and either low or regular watering. And the low watering was a press drought. So it was about 50% field capacity, which is a pretty intense drought. And because these stressors vary simultaneously, the experimental setup allowed for it to be, for the 2 factors to be crossed, so in the factorial experiment, and ended up with what was 4 environments that exposed the poppies to. This is what it looked like practically. The highlight or open light or open bench was this highlight treatment you can see here on the right, and this was just the pops were just on the bench, and whatever ambient light came in through the greenhouse, they were allowed to be exposed to. And the shade treatment is the bench here on the left. You can see they're a little bit more spaced out, and that's because we needed to make space for these shade cages that are on the top left to cover up. And these shade cages provided about 30 to 40% less light coming in through the cloth. And then within each bench, I spaced out the water treatment. So there were drought groups and regular watering groups all throughout the bench, and they were alternated. When it came to measuring traits, this was really important because traits had to be collected at specific stages to make sure we're capturing the effects of the treatments at the stage we want to characterize to later build these patterns. And the way that was done is the plants were transplanted at the same point into pots and then randomly assigned treatments, including stage. As the plants reached each designated stage, I collected data. Seedlings, when they had 3 to 4 true leaves, so that's these little leaves here on the bottom left of the diagram. In the juvenile stage, Puakala forms this rosette, which can be anywhere from 7 to 9 leaves. And then as soon as it bolted a stem, this is how we know it's transitioned to the mature stage. 2 to 5 centimeter bolted stem is the point at which I collected data on adults or mature plants. This meant that there was an ongoing process of data collection because the stages, plants were reaching the stages at different points. And that's a key difference between a study that looks at time, variation over time, and variation across ontogeny. So there was a continuous data collection as plants reached a stage. The picture here on the left shows drastic differences between drought and regular water treatments. And some of these plants were at the same stage, and you could see how different they look from having that lower water availability. How I collected traits is kind of, this makes it look like a puzzle, but really it's just a lot of fancy equipment and then not so fancy equipment in some cases to collect trait data, photosynthetic data, conductance, chlorophyll, lots of other photosynthetic parameters. We wanted to know how it performed in photosynthesis. We collected latex, prickles. We used digital software to measure leaf area. And then ultimately I harvested all of the plants to collect biomass data. I wanted to know ultimately how growth was being affected by these treatments. So it was destructive, collected all of the biomass. All right, and let's take a look at some of these results. I don't have time, there's no way to go through all the results, but here are some of the more interesting results. And we can start with chlorophyll, which is a proxy. It's effectively the greenness of the leaf, but this is a proxy for the capacity of a leaf to photosynthesize. So the more chlorophyll it has, the more possibility that it's absorbing all that light to convert it to push it through the photosystem and do photosynthesis. And so what we found was that there was plasticity at the later stages. So here in late ontogeny between juvenile and adult stages, the effects of drought seem to be very intense, where it reduced ultimately how much chlorophyll the adult plants had versus the regular plants indicated by this solid line. And then of course, plants that were in highlight, which is the solid yellow line, had more chlorophyll. But the effects of the watering treatment were already obvious here later in ontogeny in the shift between juvenile and adult. Another metric for how well the poppies are photosynthesizing is this FD over FM, which is basically the efficiency of a plant to convert captured light into energy. And this was interesting because there appeared to be no variation across ontogeny. It was just really high values from seedling to adult. And there was this uptick in the highlight regular watering treatment, where from seedling to juvenile, this like huge uptick in efficiency. But overall, there was not a lot of plasticity. This is really just showing us that under stress, Puakala is highly efficient and can still photosynthesize. So it's not a lot of effect of the treatments here. Leaf area, this is a really important trait because it tells us about how much surface area the leaves have to capture light. That will go into the photosystems to do photosynthesis and eventually grow. And as expected, the shade leaves were bigger. And you can see this is an extremely plastic trait. So as the poppies were being exposed to either low water or low light, there was a lot of variation from seedling to juvenile and then juvenile to adult as well. Ultimately, though, the plants that were under shade, which are these black lines, had bigger leaves. And this is a very well-known strategy to increase surface area for more opportunities for light capture. However, in both cases, the drought treatments, which are the dotted lines, seem to be lower and suppress growth of the expansion of the leaves. So that was something that wasn't entirely expected, but the drought treatments seem to be affecting growth even at this leaf level. Finally, we get to one of the defense traits, and I'm still analyzing latex data, but the prickles were a particularly interesting aspect of this. The prickles are a physical protection from herbivory, and it's interesting because Pukala, we don't really know what Pukala's herbivores are, so it's possible there are other functions for keeping the prickles, but ultimately what we found was no matter the watering treatment, the high light, so the open benches had more prickles, and that's these yellow lines that end up higher than the black lines. And we do see the effects of drought as well. So there's an overall increase in how much prickles, how dense the prickles are across ontogeny. But in the juvenile to adult stage, there seems to be a plateau in the production of prickles. And it's possible that this is a strategy to remain well defended under continued stress. So it's possible that that's what's happening here, and it explains why there was this uptick in production at the juvenile stage, but the transition to adult stage sort of plateaued and felt the effects of drought in the prickles. And that's a really exciting strategy there, to know that it's just planning for more stress and maintaining well defended so that the vulnerability is balanced. So this experiment sort of showed us the effects of drought and how much of a major stressor low water availability really was, especially to a plant like this that grows in disturbed areas and dry forests. And so we wanted to take a closer look at what this meant with just a focus on drought, on a drought treatment, on a drought experiment. And this is where we use the permit to collect fresh seeds, so from wild populations on the Big Island. On the top right is the map of more or less where I focus my collections, and this was all along Hawaiʻi Belt Road up into the Pu'uwa'awa'a Forest Reserve. There were other places that I collected seeds, but this is the majority of the collections. And so we went back to the greenhouse, and this time I wanted to focus on the effects of drought on gas exchange, which is a key mechanism for all plants to regulate when they have very low water. And it's thanks to these structures I'm showing here in the middle, these stomata, which when they open, CO2 is coming in and water is going out. In a stressed or in a low water environment, stomata close to reduce the amount of water that is lost. And there's a little bit of a trade-off there, because then that means there's also no CO2 going into the plant. So it's a very important trait to understand under stress. And the way we measure that is this other super fancy leaf parameter. And what this does is it measures stomatoconductance, so the loss of water in the leaf. This time around, the drought was devastating. There was very high mortality, and this was using the seeds from the wild populations on Big Island. And it was devastating. There was high mortality, especially in later stages, which could be attributed to the prolonged exposure of the treatment. And then there was one case of early flowering, which is interesting, but not completely strange, because there are plenty of plants that flower early under stress, and this is a strategy to become reproductive and reproduce before under stress to be able to propagate. And so this is just an interesting thing that happened to me in the greenhouse with just this one plant that went for it. And so when I looked at the data for stomatoconductance, the control treatment, which is regular watering, it's the solid line. It showed exactly what we expected. We expect that across ontogeny, stomatoconductance is reduced as it becomes more conservative. And that's what we see in control treatment. But the drought treatments were pretty devastating. It was very low overall, especially in early ontogeny. But then in the juvenile to adult stage, there's this uptick in somatoconductance, which could just be sort of self-regulating, and there's more tissue through which to regulate how much water is being lost. And so the values are a little bit closer later in ontogeny. And I'm still trying to figure out what this means in the bigger picture, but it's clear that water is a major stressor, even for a plant like puakala that grows in some of the drier environments. So this leaves me with a lot of questions that I'm currently writing and trying to understand what the bigger picture is of the effects here, and plans for more projects and trying to tease apart this variation. And so the take-home messages from this ultimately are that drought or low water is a predominant stressor on both resource use and defense traits. So even a plant like puakala, like I keep saying, that is distributed in some of the drier areas of Hawaii, experiences a lot of effect physiologically under drought. But it can still maintain photosynthetic performance. So there's something to be said for its for the possibility that it's engaging in some sort of resource use strategy that still allows it to grow under these stressful conditions. And then, interestingly, and as expected, ontogeny was a strong driver in that variation. So from seedling to juvenile to adults, there was a lot of variation in how these traits showed a response to this treatment. And this is, I think, where the major consideration for things like outplanting is at restoration sites. Considering how drought is supposed to become more unpredictable, more intense. This is a current map of the drought conditions in Hawaii. I took this just last night. Currently, there's a big spot of drought on Big Island, and this will expand through the summer. So people who are working on restoration sites and outplanting, knowing what the strategies are that plants use it across ontogeny, might lead to more successful outplanting. So that's something to think about, and something that I'm still trying to tease apart what works best. So in the future direction for this research, I think it needs to be expanded to the reproductive stage. So we need to understand what's happening when the plants flower, when they produce their pollinated, when they produce seed, and how well that next generation, how traits vary in that next generation, and whether or not those patterns or those trajectories can be seen in that next generation of plants as well. I think experiments with long-lived and woody species and their ontogenetic variation is important, although those are less practical. They take more time, more resources, so tricky, but I think necessary. And then looking for that genetic variation so that we can see whether there's some sort of fitness difference between the trajectories. So a plant that can regulate, or a plant that can shift from acquisitive to conservative throughout ontogeny, whether or not that gives them a better fitness for future generations. And I think with genetic variation and looking at whether or not those trajectories are inherited, we can start to figure out if that's something that plants start to figure out that contribute to the evolution of these trajectories. And then finally, application to conservation. This is very conceptual work. I had to connect a lot of dots here to sort of express how important considering stages is to when plants are exposed to stressors. But I think it's possible to use this research and apply it to conservation. And we get to know a little better how our plants are faring at the stages at which they're being exposed to these environmental stressors. So with that, I was faster than I expected. But I'd like to thank all of these people that they're instrumental in helping me do this research. And I mean, this list just grows all the time. It's impossible to do work like this on your own. And I'm really grateful to the guidance and help that all of these people in their own right helps me with. And I'm happy to take questions now. We have loads of time for questions now.
I don't have a question, but I wanted to say that your work seems to be very up and coming, and I look forward to reading more and understanding more. And I hope that we can utilize that future research for conservation and restoration projects on better outplanting of plants when the environment is becoming very unpredictable. So I look forward to learning more. We don't have any questions right now in the oh wait, question just popped up. Philip said, I have had problems germinating puakala seeds. What is your secret?
Yeah, what an excellent question. I don't have one. I just have a method that has worked for other members of the lab. It's pretty much a numbers game. So the way we germinate seeds is we do it in germination trays. And the first thing that we do to the seeds is we scarify them. So we use a razor blade to scratch the coat. Not so far that you like break into the endosperm, but like just enough to nick it. So that's like mechanical, that's like breaking dormancy in a mechanical way. We also then humidify them. So we want to like put some humidity into the seed, and then we put them in gibberellic acid, which promotes germination. It's a naturally occurring hormone, but putting it in gibberellic acid is a method that many people use to promote germination in seeds that have been in storage, for example. Even after that, Philip, I assure you that germination rates are at best 50%, but closer to like 30% germination. So when I prepare seeds for puakala, I prepare 1,000 seeds so I can have, 300 to work with. There's no secret. It's a numbers game pretty much. I hope that helps. I don't know how much that helps, but yeah.
So it looks like we have another question. How did you determine what traits to examine? That's from Brandon.
Yeah, thanks, Brandon. That's the eternal functional trait and functional ecology question, trait-based ecology question. With Puakala, what we went with was the traits that we knew were going to be essential for resource use when exposed to different light and water availability. So wanted to examine those stressors, and we looked at what are the like the effects we expected. So photosynthesis is deeply affected by light availability, especially like photosystem 2, the light reactions. And then water shifts so much along a plant's life that we were looking at gas exchange, but it also leads to carbon limitation. So we also expected changes in photosynthesis there. And then morphological traits, like I mentioned, leaf area and leaf mass per area, these are also characteristics that are traits that are affected by these factors. So, I mean, that was definitely the thought process for the physiological morphological traits. The defense traits, it was sort of an opportunistic thing because Puakala has these really cool defenses and no known herbivores, or at least none that would promote this defenses. But Puakala was documented to have like been induced into higher prickle density under high light, and latex volume also differed in induction experiments. So there's cool things there to monitor for when we reduced an abiotic factor instead of a biotic factor. And because there's no known herbivore, we were also trying to see if the prickles were having an eco-physiological function, because this plant hasn't had an herbivore in a very long time, but the prickles seem to still be are still produced, and they're produced in high amounts or in low amounts depending on the plants. Some plants don't have a lot of prickles, others do. But there was an experiment that showed that it can be induced with high light. So that sort of went with the stressors that we were analyzing. Yeah, that's a good question. Picking traits is always tricky. You want them to be the right ones to answer the question you're asking. Yeah. Yeah, I hope that helped. Thanks.
Okay, we have about 20 minutes left. I don't know if anyone has any more questions.
I breezed right through that. Yeah.
No, you did a great job. I look forward to reading more about your work.
Thanks so much, Serena. Thank you, Casey.
And if there's no other questions, we can always close up a little bit early. You do have your email if anyone needs to reach out or have any questions that they come up with after stewing on this presentation a little bit longer. Yeah. It's really just up to you guys if you want to stick around or ask more questions. We do have another webinar coming up on April 10th about Rapido death and the resistance program updates on that as well.
Who's giving the Rapido death?
Mark Hughes. So it'll be up on our website. So if you guys want to have more webinar lunchtime experiences you can always come to that one as well. But I want to thank you again, Anna, for sharing your research with us today.